Thank you very much for inviting me today. So we learn a very important lesson with Andrew today. It's essentially what's the most important thing in life. It's time, essentially. Time to play with the jigsaws, but also time with your loved one, time with your family. And artificial intelligence is really here to help you compress the time to produce output more efficiently. So this is what I'm going to talk about today. I was living in Europe, just sold my previous company, and I thought I had all the time in the world. Unfortunately, my father called me and he had the cancer diagnosis. So this is why Imagia was created in the first place, to be able to find better diagnostic tests, so we can figure out which type of disease you have, and also better treatment, better personalized treatment that will work specifically for you. So if I'm biased toward AI, it's probably because I'm very involved into artificial intelligence. We're very lucky in Canada. We have 3 of the largest AI institutes in the world, so Vector Institute in Toronto, Mila in Montreal, and AMI in Edmonton. And even greater than curiosity is to have curious mind working together. So when it comes to application, the 3 AI institutes are very focused in making a difference in healthcare. So it's an opportunity that we can make together. Are there anyone in the room that have applied AI in their daily progress or any work so far? I'm not the only one. No, there was some previous talk today about machine learning. Okay, perfect. So if you want to start doing AI, it's very important to do it responsibly and ethically. Everybody will say yes when you do that with healthcare. Of course, it's ethics first, but there's also a way to apply some of this ethical principle by design. So I'm going to talk to you about those kind of principles. Encourage you to read this Canadian Declaration of AI. So it's called the Montreal Declaration of Responsible AI, 10 principles. There's about 40 declarations in the world, and this is probably one of the best crafted from an overall study. So if you're not familiar with AI, the recent technique that really made the headline around 2012 was called deep learning. So there's this famous book called Deep Learning that you can read, authored by Jan Goodfellow, Yoshua Bengio, and Aaron Corville. So Yoshua Bengio is the founder of the Mila and Turing Prize Award. What's interesting about deep learning technique is, for this particular example, they use it to do natural language processing. So the technique itself really allow you to understand free text at a conceptual level. So they use the same deep learning concept to translate their book automatically in French. So you have it also in French, l'apprentissage profond. And that's very interesting, because when you are in healthcare and you see a lot of free text reports, even from hospital to hospital, in the same language, the way that they're written will differ, and you want to understand the conceptual that underline those reports. So there's very good thing to be able to do from a natural language processing. For this particular example, it worked about 90%, so that's why they still continue to advance AI. So today I'm going to talk to you about something that is, I would say, very narrow, but at the same time that have a lot of application, and I'm going to lay a term forward called digital biomarker. So you're very familiar with biomarker. Digital biomarker, it's essentially the same, but just looking at the data. You're looking at essentially 2 types of data that sometimes are not related, or they are, but you don't know why. So for example, a genetic mutation and a response to a specific treatment, why they are linked together. So that might be a digital biomarker. So I'm going to provide to you some example of digital biomarker and how they can be of help in the healthcare environment. So 3 types of AI biomarker. The first one on the top left is really called a detection biomarker that has been around since 1998 in the computer-aided design, computer-aided detection system, where essentially you just want to detect an anomaly. So an example, you can detect a nodule, but you can also detect an anomaly in maybe in a genetic sequence or in a text or in lab analysis. That's very interesting to make the difference between normality and an anomaly, but that will not very revolutionize the way we conduct healthcare today. What's really interesting, and I think what's going to interest you more, is how can we use the predictive power of AI to essentially predict what will happen next. So artificial intelligence is excellent at looking at different longitudinal time series of data, like your health, that will evolve over time. And data points can vary from specific genetic mutation to images data to lab result to treatment and outcome, and they will evolve over time. You want to make a sense out of that. And you can use those digital biomarker just looking at data and predict what will be the result of an expensive, an invasive, and a time-consuming test. So all going back to time. So essentially, you can look at an image and predict the result of a pathology analysis. You can look at an image, and you would see in a low-dose CT scan probably some macroscopic effect of a gene mutation. So of course, if you want to confirm that, maybe you will just do that specific gene test. So we see a lot of improvement essentially in triggering those more invasive, costly, and tests that sometimes are triggered unnecessarily, but also most of the time they're not triggered, and they should be. And finally, where the moonshot is on the reveal part is really to predict tests that does not exist and probably will never exist in the physical world, like predicting response. So if you look at a lot of patient data, you might be able to predict the response to a specific treatment. And I'm going to give you example. So I'm going to start to give you an example by simple one, like predicting pathology. I think I have a short video for that. Not sure if the sound will work.
So I want to demonstrate the AI program in action. There's a very small polyp here, a couple of millimeters in size. In white light, it looks like it has a little bit of a central valley sign in it. I'm going to turn the close focus on, and then to activate the AI, all we need to do is turn on the narrowband imaging. You can see that the program is really immediately developing high confidence that this is a type 2 lesion. It'll do it either in the near focus, it'll also do it in standard focus from a little greater distance. So this would be a great candidate if we actually could perform, resect, and discard. This lesion has basically 0 chance of having cancer in it. We could remove it and throw it away. But it demonstrates nicely the performance of the model with a high confidence diminutive type 2 lesion. With the sigmoid colon now, again, still with our pediatric scope. We've got about a 5 millimeter lesion. We'll take a close look at it. And the program is telling us that this is a type 2 lesion. I'm just going to try to get in here close to verify that that's the case. I do think that the surface pit structure is primarily tubular, and yeah, definitely a type 2 lesion. The program knew that before I did, but...
I'm going to stop the video there. So I really like this sentence of one of the most famous key opinion leader in colon cancer, where he said the machine saw it before I did. And essentially, that's an example in a medical device environment, but you can clearly see I was going to lead that to other example, mostly touching on the pharma side. So The question is, how do you do those type of digital biomarker that will be able to predict response, that will be able to predict the result of a genetic test or pathology just by looking at the data that you have in your institution. And the response is, the more and more we understand specific disease, the more and more they get specific. And the less patients you have that have this specific disease in your hospital, in your environment. And to do artificial intelligence, working with 30 patients is not that great. We've done it for a specific study, but you need more. So you want to be able to look at multiple data from multiple institutions across provinces, across countries. And that's become a challenge, because if I go to a hospital and tell them, oh, can I please have access to all your data? I'm going to export it out of your hospital. That will never work. So we've taken a different approach where the data stays where they are. And it's essentially the artificial intelligence model that will go from one side to another, or will learn from a federated approach. So the data doesn't move, it's just the learning that gets exchanged in a way. And that was the beginning of about a year ago, and we have some very strange old version of Office, but there was a lot of interesting text here that I'm going to explain to you. But at least you can see the Terry Fox proposal that we made to the federal government. So a year ago, federal government asked for how can we build a national platform to be able to foster new digital discovery. There was about 40 consortium applications, including the one from Sunnybrook and the one from Terry Fox and Imagia, with about 100 members. And we were both selected as a winner of this Stream 4. So for the Terry Fox and Imagia co-led application with many hospital partners, the first time that the AI Institute worked together, we will be able to deploy this discovery platform. And I'm going to go into a little bit more detail on that platform in multiple regions across Canada. So those digital biomarkers can be found and put to patient. So let's dive into one of the projects of this platform called Evidence. So this is an Imagia product. Essentially, what you want to do, if you start from the top left position, you start with the clinicians and the patient. Everything is around the patient. The patient provides the consent, patient generate data, and patient has specific disease, specific treatment, and specific response. So you want to, as a clinician, generate your research project. So what's the first thing you do is you build a cohort of patients. That takes a few months. I've been into a hospital where they take like spreadsheet and Excel, and they find the patient for their specific research, because essentially it's not a keyword search. You have specific inclusion and exclusion criteria that are complex, that are conceptual, like pseudo-response. There's nothing such as pseudo-response code or in the data explicitly. You need to explain what do you mean by pseudo-response. The definition might be different from one researcher to the other. So rather than taking 9 months to do that or 6 months, we've automated that with artificial intelligence. So think of AI not as the end product. Think of AI as a tool to help accelerate your research and save you time. So the first step, where you see the cohorting part, we help that with an active learning to understand what the clinician means by their inclusion and exclusion criteria. The second part of the step one, where you see A and B, when you do AI, you want essentially the model to understand the difference between, let's say, two cohorts of patient type A and patient type B, or the link between those two types of data. Maybe one of them is cancerous and the other one is cancerous but from another type. Typically, an AI model will need label. and label are done by experts. And those labeling techniques introduce errors because nothing is perfect. So we've replaced part of that with AI machine and input from the experts, whereby if you ask the question to two experts, the difference between their answer would be minimum. So you're able to measure the bias that you introduce to the data, and the objective is to bias the label as least as possible, because the data that you're working on already contains error from the beginning. So the output will never be perfect, but you want to have a process that is as robust as possible. So at the end of step one, you have a nice cohort of data that contains label and that also have the proper metadata for learning. You would say, what's metadata? Essentially, when you conduct research on a traditional way, they might have some acquisition parameters that are not very important, because when you look at the data, they look the same. But in some equipment, you can tweak the parameter, or when you do gene sequencing, you can change a little bit the acquisition or the protocols. That might have some subtle differences in the data, and that is very important for the algorithms to know what were those metadata. Step one is done almost automatically. Few days, few weeks, you're done instead of few years sometime. You go automatically to step two, where the algorithms will try to find, is there a weak signal? Is there a signal between the two types of data that you're trying to differentiate to build out that digital biomarker? And if there is, then we can validate that in other institutions. We can validate the digital biomarker on other retrospective data. You can also conduct this discovery with ethics board approval simultaneously from multiple hospitals in a federated approach. And what's important, you want to make sure that once there's a digital biomarker, and a good example of a digital biomarker might be a companion test, but a digital companion test. So if you figured out a drug along with the clinical trial, there could be a build-up of a digital companion test that could trigger the prescription of your drug, that could orient the patient stratification while you're conducting the trial. So you want to make sure that this digital companion test will go to market. So an ISO 1345 company, we can make sure that this will be licensed out properly as a medical device, as a software, to either medical device or pharmaceutical companies. So an example of radiomics or radiogenomics. Anybody in the room are familiar with those terms? Perfect. So essentially, radiomics and radiogenomics is a science that appeared about 10 or 15 years ago, but with AI is getting more and more attention, because essentially, just by looking at images, you can see macroscopic effect of gene mutation. So that's very interesting, because you can see if those changes occur because you're doing the treatment, you can measure the response much more effectively. So here we have two types of cohorts, one that might have a specific gene mutation and the other that don't. And you want to see from the images what are the features that do link the image to the gene, the specific gene mutation. And those are not one feature. There will be a lot of features, and then you can find if there's expression in the gene in the image. Some example of the platform. So we really want to empower researchers and clinicians that are very good at what they do, but they're not AI experts. Very bright, but they don't want to spend 10 years learning AI and doing that in the weekends. So they can use the platform, see maybe links between images and text with color coding. We try to be as intuitive as possible to work with that platform. So now I gave you an example for application in a medical device, real-time, live inpatient, going into specific application for lung cancer. In this specific example, I'm going to start the video. Here we We're talking about lung cancer, non-small cell with the EGFR mutation, one of the most prevalent mutation in lung cancer. There are specific treatment that works better for EGFR positive patient, and unfortunately, those treatments are triggered too late into the process. A patient that have more than 5 millimeter nodule will tell that person come back in 3 months until the nodule doubled in size, and by that time it might be 6 months or even more. With that kind of techniques, we're able to pick out automatically the malignant nodules, and the next step is to pick out automatically the nodules that have specific mutation. What's really interesting about this AI prototype here is that the prototype was trained by showing 2 types of images. Images that contain nodules that were benign, and images that contain nodules that were cancerous. We didn't tell the machine which image was which. You have special technique to do what we call unsupervised learning, and the machine was able to generate a representation of all the features that represent what is a malignant nodule. So that's very interesting because now we have characteristics that are unbiased that represent malignancy. So going the next step, we will be able to do that from an EGFR perspective. The literature is there. Is now we need to build a commercial product out of that. Here's another example. Can anyone in the room tell me what this is? Please. Sorry? Lower. So it's a CT, actually it's not a CT scan of a lung. That was a trick question. That's a computer-generated CT scan. That's essentially a dream of a computer into which we shown a lot of normal CT scan. And we asked the machine to output the feature in a graphical representation. So what you see is a normal CT scan generated from the computer. So you would say, oh, that's cool, but what's the purpose? The purpose is if you understand very well the normality from a feature perspective, detecting abnormality is a much more easier task. And then the next step, if you understand abnormality very well from a list of features, detecting specific pathology, and then detecting some specific mutation, or even then specific response rate, becomes much more easier. So we're starting to enter a world where we almost go in a hypothesis-free research where we show data to the model and we ask the model, can you show me the list of features that links those 2 data together? What's really important from an AI perspective when you build a model is to be able to differentiate when the model is certain or uncertain about the categorization of the class. So let's say if you want to build a binary classifier, cancer or not cancer, in the end you should have 3 classes. Yes, no, and I'm not sure. So those are the yellow line here in the middle where you might want clarification or input from the expert. So we're really playing hand by hand, machine, human together to make sure that we can increase the accuracy either in a training phase or while we use the thing. I was talking a lot about features that do characterize the link between 2 types of data. So let me give you an example of how we can represent those features. So in the top part, you have a nodules, type of nodules that have low survival rate, less than 3 months. And in below, you have other type of nodule that have higher survival rate, more than 12 months. And we show that to the algorithm, and the algorithm we ask, can we see all the features that do correlate between survival and type of images? And there were like 3,000 of those features. Some of them in 3D, human perspective, human eyes. It's very difficult to understand the pattern, but the machine are able to really characterize some of the patterns. And if you rank them in a log scale from the least important to the most important one, so if you look at the red circle, there were 3 features that are significantly more important than the others. One of them is linked to the heterogeneity around the tumor that is predictive of a low survival rate. So the machine did find that automatically. We already knew that, but the fact that it found that automatically is an excellent news. And the 2 other, of course, are very interesting from a pharma perspective because it's a new feature, new way of thinking, why there's low survival, why there's super responder and non-responder. And those kind of, I would say, we're starting to go into the explanation. We talk a lot about AI as a black box. It's not a black box. It's really a transparent box where you can connect and list the features. Some of those features can be explained. Some other are more statistical pattern, but we can list them certainly. So I think this is a very positive outlook in terms of hypothesis-free research. So thank you very much for listening to me, and hope we'll enjoy next conversations.